Welcome back to the second session of this afternoon. This is the second part of the Japan-Taiwan Bilateral Symposium, and we will have three speakers. The first is Professor Xiao Yuan from National Ming University, but he's in the Jiaotong campus. He has been an outstanding structural biologist focusing on the DNA-protein interactions. And today he will be talking about the story about APE1. So Professor Xiao, the floor is yours.
Good afternoon, everyone. I am Xiao Yuan. I am so happy to join the APA, APPA, and TBS Joint Congress 2022 on site because I can see a lot of old friends. So it's my pleasure to be here to show you our current result. And today I would like to talk about a famous DNA repair nucleus named APE1. APE1 can process its duplex DNA substrate in both endo and exonucleus membrane. Interestingly, the substrate specificity of APE1 in endo and in exonucleus membrane is quite different. So it is very interesting to understand how APE1 uses the same active site to process different substrate in the different membrane with different substrate specificity. Based on our structure and biochemical studies, we established a model named induced space filling model. This model it can to elaborate the substrate selection mechanism of APE1 in both in endo and exonucleus membrane. And the study is also very important to reveal the cellular function of APE1 in various DNA repair, and also very important to understand the mechanism of drug resistance related to APE1. So I will show you the detail later. Before talking about APE1, please allow me to introduce my laboratory briefly. My lab is a structural biology laboratory, and we use the structure and biochemical approach to understand the catalytic properties, substrate selection mechanism, and cellular functions of nucleic acid binding proteins. And currently, we are engaged in the nucleus involved in the immune regulation, such as SAMHD1, RSH2, and TREX1. Mutations in those nucleus leads to a serious, severe autoimmune diseases. So we are very interested in how those nucleus are involved in the innate immunity regulation. And we also want to know the relationship between those nucleus and autoimmune diseases. And our target APE1 is also the component of ER bond set complex. It works with TREX1. On the other hand, when the cell stressed by genotoxic stress, APE1 and TREX1 and their working partner translocate into the nucleus and participate in various DNA repair pathways. So we also aim to understand the role of those nucleus in the DNA repair. Interestingly, the nucleus we focus on, they are also the drug target. So we also aim to develop their inhibitor and apply it in the anti-cancer therapy by structure-based drug design. So here is the brief introduction of my laboratory. So let's come back to the APE1. APE1 is a multifunctional protein and composed by N-terminal redox and C-terminal nucleus domain. The C-terminal nucleus domain is with both endonuclease activity and exonuclease activity. The endonuclease activity of APE1 is associated with base excision repair, and the exonuclease activity of APE1 is related to NIR and the removal of damaged DNA base. Both activities are essential for maintaining the genome integrity of cancer cell. So APE1 is also a very good drug target against tumor. So scientists develop a lot of APE1 inhibitor, especially the nuclease inhibitor like gasposol, luciton, and LODDC. And they already apply in the clinical trial in different phase to against the cancer. So now we can understand APE1 is exonuclease and endonuclease, and with the biomedical importance. As I mentioned earlier, APE1 is endonuclease and important in the base exchange repair. The BER is a DNA pathway to correct individual oxidative DNA damage. At first, the DNA glycosylase can recognize the damaged DNA base and broken the bond between the base region and the ribose region. After that, this structure is called AP site structure. And APE1 can specifically recognize the AP site structure and make a nick at the phosphodiester bond here. And after that, DNA polymerase beta and other protein can complete the DNA repair progress. And here is the APE1 AP site containing double-strand DNA structure. The structure clearly shows how APE1 recognizes the AP site containing double-strand DNA and how the APE1 nick the phosphodiester bond here. So we already know the endonuclease activity of APE1 is well studied. Besides the endonuclease activity, in 2000, APE1 was reported as a 3' to 5' exonuclease and without base preference. And this gel-based experiment can prove that APE1 is an exonuclease. Here is the uncleaved double-strand DNA substrate, and we label the fluorescence dye at the 5' end. When we mix the DNA substrate with APE1 with different concentration, as you can see here, there are more later band formation along with the increased APE1 concentrations. So this experiment show APE1 is a 3' to 5' exonuclease. So if we synthesize the damage base at the 3' end, like A-oxyguanine, APE1 still can remove the damaged DNA bases with similar efficiency to the normal DNA base. So we can conclude that APE1 is a 3 to 5 exonuclease and without base preference. And the exonuclease activity of APE1 is also associated with drug resistance. As we know, some nucleotide analog like AZT or LODDC, those nucleotide analog can incorporate into the DNA to block the viral or cancer cell DNA synthesis to kill them. But the virus or cancer cell can use their APE1 to remove the nucleotide analog and induce drug resistance. So the exonuclease activity of APE1 is a very important project and need to understand the catalytic properties. Besides the base preference, APE1 seems to have the structural preference. And you can see here, when the substrate is a duplex DNA with perfect pair at the terminal or with the mismatch on the terminal, APE1 prefer to process the mismatched DNA than perfect pair DNA. And ability of mismatched DNA processing of APE1 is used to work with the aeropole polymerase, DNA polymerase beta, to free the newly synthesized DNA. And even the ability of APE1 in process matched double-strand DNA is weaker than processing mismatched double-strand DNA, but the ability of matched DNA processing is also very important. Because the ability is associated with nucleotide incision repair, NIR, trinucleotide repeat expansion related BER, DNA single-strand breaks, and apoptosis. However, the actual rule and the natural substrate of APE1 in those DNA repair pathway is remain unclear. And the final structural preference of APE1 is showing here. As you can see, if the overhang region, 3' overhang over longer than 2 nucleotides, this kind of substrate is not good substrate of APE1. So in general, one DNA repair pathway requires many different nucleases with different structural preference. They can process different intermediate DNA to complete the DNA repair progress. So a very good way to understand the rule of APE1 in those DNA repair pathway is try to understand the structural preference of APE1. So we try to synthesize many different DNA intermediate like nicked double-strand DNA, gapped double-strand DNA, 3' flap double-strand DNA, and 5' recessed double-strand DNA, and with or without a mismatch in the terminal as a substrate to reveal the rule of APE1 structural preference. So I will show you our result later. So we have two specific end. So the first specific end is we want to understand the structural basis of the base preference of APE1. While in the endonuclease member, APE1 can specifically recognize the AP site structure. But in the exonuclease member, APE1 without any base preference. This phenomena is associated with the drug resistance related to APE1, so it is a very important question. And our second end is we want to use the biochemical and structural study to understand the structural preference of APE1 and try to reveal the rule of APE1 on various DNA repair pathways. So in order to reach our end, first we try to determine the APE1 double-strand DNA complex structure. So as you can see here, we try to determine the complex structure at the 2.7 resolution. As you can see here, APE1 binds to the double-strand DNA in a terminal binding mode. APE1 specific interact to the terminal of the duplex DNA. And the structure is very useful for us to understand the catalytic properties of APE1 as an exonuclease. In the structure, APE1 cleave the last nucleotide at the 3' end. So this structure is a product complex structure. And after the RNA binding, we can see here is a bridge-like structure formation. And the bridge-like structure is composed by a methionine and arginine residue. So we name it RN bridge. And the bridge-like structure only can be observed in the DNA bound complex structure and cannot observe in the APE1 structure, which is proved by structural alignment. And here is the section view of APE1 structure and the APE1 DNA complex structure. Here is the active site. As you can see here, the active site is opened. But when RNA bridge formation and the DNA bind to the APE1, you can see here is the RNA bridge. RNA bridge is filling the space of the active site and make it become smaller and become a channel-like structure. And the RNA bridge can use to unwind the DNA base pair at the end of duplex DNA. As you can see here, the RNA bridge separate the two DNA strand and make the sister strand into the active site. And the last nucleotide at the 3' end lay on the hydrophobic pocket. And the RNA bridge mediated DNA cleavage we name and use a model to describe. The model called induced space filling model. And the induced space filling model can use to explain the base preference of APE1. So in the endonuclease member, you can see here, the RNA bridge insert into the duplex DNA and between the open base and the AP site. So the structure like here. After the insertion, you can see here APE1 is flapped out to the active site and fit very well to the product pocket. And as you can see here, here is a steric hindrance. If there is a normal DNA base, the space is enough to put in a normal DNA base. So in the endonuclease member, APE1 specific target to the AP site structures. When in the exonuclease member, APE1 bind to the terminal of duplex DNA and separate the last base pair. As you can see here, the last nucleotide at the 3' end can put into the active site nicely. And because there are no upstream phosphate, so the base can lay on here. In addition, the product pocket is composed by many hydrophobic residues like phenylalanine, leucine, and tryptophan. So they cannot interact to the base by specific hydrogen bond. Therefore, when AP1 is an exonuclease, is without a base preference. So based on our structure, we reach our specific N1. We reveal the structural basis for nucleotide specificity of AP1 in both endo and exonuclease member. And next, we will talk about the structural preference of AP1. As I mentioned in the introduction, when the substrate is a double-stranded DNA, APE1 prefer to process mismatched DNA than the matched DNA. And the 3' overhand longer to 2 nucleotides, not a beta substrate of APE1. Try to understand the structural preference of APE1 more clearly. We also synthesize recessed double-strand DNA like this, and gapped and nicked double-strand DNA as substrate. And those substrate also with the match or mismatched terminal. In general, as you can see here, APE1 still prefer to process mismatched DNA than matched DNA. But if the DNA is with very long 5' overhand, like the recessed DNA, or the DNA with the flank region, like gap DNA, the digestion efficiency is very similar between the matched and mismatched DNA substrate. When we use the gap or nick DNA as a substrate, as you can see here, APE1 prefer to process gap DNA. The activity of APE1 on nick DNA is very weak. So after the systematic analysis of our data, we can conclude 3 rules of APE1's structural preference. The first one is APE1 prefers short 3' overhand. The nucleotide should shorter than 2. And APE1 prefer to the DNA with longer 5' overhand, like a recessed DNA, or the DNA with the flank region, like a gapped DNA. And the third rule is APE1 prefer the gap size bigger than 1 nucleotide and not prefer to process nicked double-strand DNA. And our induced space Bellum model also can use to explain the structural preference of APE1. So the first one question is, why longer 5' overhand substrate is better? So as you can see here, here is a lot of arginine and lysine residue, and those residue can interact to the 5' overhand region and the flank region of the substrate DNA. So after the hydrogen bond formation, catalytic difference between matched and mismatched DNA can eliminate. And this is why the longer 5' overhand substrate is better. And the second question is why 1 NT long 3' overhand is better. It is because after the space filling, the active site becomes very small, only can contain 1 nucleotide. So our structure can explain why 1 NT long 3' overhand is better. And the third question is why APE1 prefer gapped DNA than the nicked DNA. It is because when in the nicked DNA, here is the upstream phosphate, and here is the normal DNA nucleotide. It is difficult to put those 2 components together into the narrow active site. You can see here is a steric hindrance. So in order to prove our structural observation is correct, we try to synthesize a nicked DNA without the upstream phosphate. When we use this substrate in the nucleus activity assay, as you can see here, the cleavage pattern is much better than the normal nicked DNA. So this experiment strongly support our hypothesis. And the last question is, can our induced space filling model can use to explain why APE1 prefer to process mismatched double-strand DNA? So in order to answer the question, we try to superpose our APE1 dsDNA complex structure to the previous DNA complex structure. As you can see here, the structure is published at 2018. So when APE1 binds to the duplex DNA with mismatched, as you can see here, the flank region is bent and move about 10 degrees. The moving can avoid the structure hindrance between the phosphate and the DNA base. So we can conclude that here are two different binding modes exist when APE1 binds to the perfect pair DNA or bind to the mismatched DNA. So in order to prove the two different binding modes exist, we try to use the Joule-Shift assay to prove. So if your substrate is AP site containing double-strand DNA or perfect paired gapped DNA, you can see two complex bands. The first complex band is a low molecular weight protein DNA complex band, and another band is high molecular weight complex band. But when the substrate is mismatched DNA, we only can observe the high molecular weight band. It means that the binding mode is different. The experiment proves our hypothesis indirectly. So finally, when we understand the structural preference of APE1, we can try to understand which intermediate DNA probably is the natural substrate of APE1. So we can give a hint of the role of APE1 in those different DNA repair pathways. In summarize, our study can give us a very good answer of APE1's base preference and structural preference. We can use the structure and biochemical study to reveal the structural feature of APE1's base specificity and structural preference of APE1. And this study is very important for reveal the cellular functions of APE1 in various DNA repair pathway like shown here. And our study is also very important for understand the mechanism of drug resistance related to APE1. And all of our data have already published in the following papers. So if you are interested, you can try to read the two papers. And in the end of my talk, I will say thank you to my financial support, especially the most young scholar fellowship and the research center of IDS2P in National Yang-Ming Jiao-Tong University. And all the data in our study are collected in the National Synchrotron Radiation Research Center. So I will say thank you to the NSRC. And I also want to say thank you to the member of my laboratory, the PhD student Dong Zhang, and the master student and Kai Wei, and my old student Kai Chen is here. They complete the APE1 studies. And here on the QR code, you can try to screen the QR code. The first QR code is my lab website. The second QR code is my lab's YouTube channel. We upload several lectures related to structural biology and put some very nicely PyMo movie example in our YouTube channel and some research in this channel. So try to screen the QR code and subscribe to our YouTube channel. Okay, I will end here and thank you very much and welcome to take questions.